Good evening, everyone. Welcome to the Week 10 live interaction session for the course Fabrication Techniques for MEMS-based sensors from the clinical perspective. So this week, first we will start with a recap of the content. Okay. Then we will move on to some problem questions. So this is about me. And every week on Monday from 6 p.m. to 8 p.m., we'll have this live interaction session where we will discuss some problems regarding the content that was covered in the uh, corresponding week. And this is the meeting link. And here are the links for the channel as well as the uh, uh, Google Drive where the videos and the slides will be uploaded. In case you have doubts, you can reach me at this email. So in week 10, the content that was covered was regarding the introduction to clean room and clean room techniques. So where we saw about the various equipments used in clean room and the gowning procedure. And we also had an introduction to equipments such as impedance analyzer, DAQ, function generator, and so on. Okay. So now we can start with the problems. Problem 1. The type of incubator used for cell culture that can provide controlled environment for culturing is option A, O2 incubator, option B, CO2 incubator, option C, CO incubator, option D, CO2 O3 incubator. So, which is the correct answer for this question? So, the correct answer for this question is CO2 incubator. So, the correct answer for this question is CO2 incubator. CO2 incubators are enclosures that are used to precisely control the environmental variables that is needed to grow any biological samples or do any type of cell culture. And if we look at what are the conditions that are required to maintain the same environment as that of like human is that we should have a neutral pH, and then the body temperature should be maintained at 37 degrees centigrade, and also the relative humidity should be greater than equal to 90%. So these are some of the conditions that have to be maintained for the human body, like to have some same conditions as that of the human body conditions. And so these are some of the conditions that were made in the human body conditions. So for that reason, we use this CO2 incubator. Why do we? Also there is some particular amount of carbon dioxide concentration that has to be maintained. Like, why do we need like a particular carbon dioxide concentration? That is because if we want to grow cells in the cultured medium, I had mentioned that there are three important main things that like conditions that we have to maintain so that like we get the in vivo conditions. So one is the pH should be neutral, right? Then second one is the temperature that should be same as the human body temperature, which is 37 degrees centigrade. And the third one is that the humidity also should reflect the human body humidity condition, which is around greater than 90 percentage humidity. So since we want these three kind of conditions, we need to have the incubator. And like generally the CO2 incubator that we are using, it will have carbon dioxide in the concentration of 5 to 10 percentage. Why do we need this 5 to 10 percentage? This is because we need to make the medium grow in neutral pH condition. And what will happen is, if there is too much CO2, what will happen is that cells will have water content in it, right? This water will get combined with CO2 and it will form carbonic acid, which is H2CO3. And also it can form some kind of bicarbonate as well, which is HCO3 minus. So like we know that bicarbonate is it is a base, whereas carbonic acid is an acid. So since we have like combination of both, ensure that the pH remains somewhat close to neutral conditions. So by like adding or by maintaining the carbon dioxide at a particular level, we can balance the oxygen that is present in the like in the particular environment, and we can prevent the cells from becoming either too acidic or too alkaline. uh, this is why do we need that? This is because we want to closely mimic the human body conditions, and also when the cells like are kept in an acidic or alkaline environment, the cell growth will get inhibited. So that's why we are using a CO2 incubator. However, if we are seeing like CO2 levels of human, it is around 400 ppm, which is 0.04 percentage. So that's why we go for a CO2 incubator, because we want main reason is we want to maintain a neutral pH for the cell culture. Is this clear? If this is clear, we can move on to the next question. Every person working inside a class 100 or class 1000 clean room must follow PPE protocol. PPE stands for option A, proper personal equipment, option B, personal protective equipment, option C, personal protection enhancement, option D, possible protective equipment. So, which is the correct answer for this question? You would have heard like for in order to like before we go inside a clean room, we should wear some like some kind of PPE, right? And this PPE is generally used in semiconductor clean room, but you will find its use in various fields like medicine and surgery as well. So what is the expansion for this PPE? So the correct answer for this question is option B, personal protective equipment. Is meant to protect the person. Okay. So here you can see a person wearing a PPE. So this white color suit is called as a bunny suit or a protective overall. And you can see that this suit will have a hood that is attached to it. Okay. Then you can see that this person is also, and he has some like protective boots over the overall. Then underneath this bunny suit, what he will be wearing is he's also wearing protective glasses or clean room glasses, and underneath he'll be having some additional protective gloves or some additional gloves, medical face mask, and like a hair cover. So these are all the things that will form the part of the pers personal protective equipment that is used in clean room. And one of the main reasons why we have this protective overall is to minimize the amount of particles that are being shedded by the human. So generally, humans will shed lot of particles, and when they move, when they walk, they will shed a lot of particles. We want to minimize the amount of particles in a clean room, right? So by class 100 and class 1000, we are telling that we have like certain restrictions regarding the number of the particles as well as the size of the particles that are present. So if you want to minimize, we have to ensure that whoever is coming will have will not expose any skin, which will from skin we can have many particles which will shed. So that's why people wear this bunny suit. And also not only just for reducing the amount of particles shed by a person. When a person is in clean room, he will work with chemicals and so on. So in order to protect the person from harmful chemicals, exposure to those chemicals by accident, he will also wear mask as well as the protective glasses and gloves. So is this clear? If this is clear, we can move on to the next question. Problem 3. Which of the following statements is or true? Option A, keep the UV light on while working cabinet. Option B, ensure hands are washed before and after using the biosafety cabinet. Option C, disinfect the surface of the cabinet before use. Option D, disinfect all the items that are taken in and out of the biosafety cabinet. So which ones are the correct answers? So this is related to a biosafety cabinet. So this problem is related to the use of a biosafety cabinet. So there are certain rules that have to be followed. Right. So the correct answer, and like this is a multiple option, uh, multiple correct option question. So which one is the correct answer for this problem? Which of the following statements is are or true? A biosafety cabinet is something it is kind of a containment device for biological material, and so we will keep the biological materials inside that particular cabinet. It is like how we are using fume hood while handling hazardous chemicals. Okay, so we have to ensure that before we are using the biosafety cabinet and after we are using the biosafety cabinet, that are so option B is correct. We'll come to option A later. Okay, then option C is disinfect the surface of the cabinet before use. So before using, we have to ensure that the surface is clean, and also that there is no other contaminants that is present or at the uh, surface. So this is also correct. Then we also have to disinfect all the items that are taken in or taken out of the biosafety cabinet. So that also is correct. Now let us look at option A. So we have in option A, it is mentioned that we have to keep the UV light while working in the biosafety cabinet. Okay, why do we need UV light? So this UV light is like it is generally act as a germicidal lamp. So it is to keep the interior clean when we are not using. Like we don't want to get exposed to this UV light. So this is generally used not using. Okay. And it is not required that we have to turn on the light for a long time, like 15 minutes, like a time period of 15 minutes should be sufficient, because like it will make sure that all the germs are killed by that time. And also like UV light can also cause degradation of certain materials, and it is like harmful when like if there is any some gas burner or something inside the biosafety cabinet. Okay. So the correct answer for this problem 3 is option B, C, and D. In B, we are making sure that option A is wrong, because like we will use the UV light to uh, kill the germs when we are not working in the biosafety cabinet, not like when we are working in the biosafety cabinet. So that is the one thing that is wrong in problem 3. So options B, C, and D are correct. Hmm. Is this clear? And here in this figure, you can see like 3 different kinds of biosafety cabinets, class 1, class 2, and class 3. Uh, so these biosafety cabinets depends on the biosafety level. So class 1 and class 2 biosafety cabinets are used for bio 1 and 2. So these are used for biosafety levels 1 and 2. But like class 3 is generally used for biosafety levels 3 and 4. So like here the number increases, like the hazard will also increase. One more thing is that biosafety cabinet will provide protection to the sample, to the person who is working on the sample, as well as to the environment. So it will have 3 functions. And like so some organisms can result in the formation of some kind of aerosol, which is harmful when ingested, and it should not get into the environment as well. So for that purpose, okay, is this clear? We can move on to the next question. This is problem 4. Uh, Using the wet bench, if you are unsure of the chemical discard procedure, what should you do? Option A, refer to the material safety datasheet, which is in short for MSDS. Option B, pour into the drain. Option C, on the bench unlabeled. Option D, try adding water to dilute the chemical. So which is the correct answer for this question? So which is the correct answer for this question? Okay. So the correct answer is option A, refer to the material safety data sheet. Okay. This is because like certain chemicals should not react with other chemicals. They may like produce certain violent reaction. For example, like acids and solvents should not get mixed. And also, if you see solvents, like solvents has this tendency to get combust, like they can get combusted easily. So when we are disposing solvents, like we do not just dispose them into a drain. We'll just like get keep, keep them disposed in a particular container so that they can be disposed like. And also, like it is not good to leave it on the bench unlabeled, because if you see most of the chemicals, they are like transparent. They do not like they look kind of look like water only. They do not have any distinct color, except like some of them may have a distinct color, but most of them like they look like water. So we should not leave it on the bench unlabeled. And like the fourth option is also wrong, because like say there is a general rule while handling acids that we should add acid to water, not we should not add water to the acid. Say, let's say if you're having a petri dish with acid, and if you add lot of water, then like the acid like in some cases like exothermic reaction kind of happens. So that is bad. That's why we try adding acid to water, water to acid. So in the chemical, in the material safety data sheet, they will generally provide all the details regarding like what are the disposal procedures and so on. So here you can see some of the sections that are present in a material data sheet. So it contains the information about the product, the name, the manufacturer's name, the supplier's name, and so on. Then also it contains the information regarding the hazardous ingredients that may be present in that particular product, and the physical data, fire and explosion data, reactivity data, toxicological properties, preventive measures, first aid measures, preparation information, and so on. So these are all the things that are generally present in any hazardous or non-hazardous material data sheet. Also, like here you will be from the reactivity data, you will kind of know which substances you should not react. So that will give you an idea of how you have to dispose as well. So let's say, like I mentioned before, like solvents and acids, like people don't generally mix them while discarding. So that like you will kind of come to know about that from the reactivity data. So it may react with certain chemicals and give a violent reaction. So we don't want to dispose them together. So we will make sure that we dispose them in separate manner. So to summarize, like discard procedure, like can sheet, because it will give you all the details regarding the chemicals. So like how it will react with other chemicals and so on. Based on that, you can determine what will be the safest way to discard that particular chemical. Is this clear? If this is clear, we can move on to the next question. Which among the following holds true? Option A, PPE depends on the process handled. PPE can be worn and stored only inside the cleanroom gowning area. Option C is contaminated PPE should be washed or discarded accordingly so that the next user does not come in contact with it. Option D, all of the above. So we already saw in previous question that PPE is personal protective equipment. Which is the correct answer among this following? Okay. So first, let us look at the options one by one. First is PPE depends on the process handled. Right. This is correct because there are various processes that we do in the clean room. If you are doing some sort of process which is having like chemicals involved, then you should ensure that you are like wearing the additional gloves that are required. So let's say if you are doing any process with wet bench means like you are doing any process with wet. Then in that case, you will have to wear additional gloves, then sleeves, protection sleeves, uh, an apron, and also like some like there are special gloves for handling acids like Eptalon gloves and so on. So those gloves also you'll have to wear additionally while handling the chemicals. In addition to that, you will wear a head shield as well, head shield or protective goggles. But when we are doing some dry etch process and so on, we do not need like the protection sleeve or head shield or protective glasses. So the PPE will depend on the process handled. So this is correct. Okay, next option B is PPE can be worn and stored only inside the clean room gowning area. So that is true. And in clean room, the reason that we are wearing PPE is mainly because we want to reduce the like generation of person. So when a person goes inside clean room, we want to reduce the amount of particles that he is generating. And also like in order to minimize like the amount of particles that will come when the PP is taken outside the clean room area, we will store and wear the PP only inside the clean room gowning area, which will also have some particular like particle control. So it will not be like environment, it will have some kind of particle, it will have less number of particles. Then option C is contaminated PPE should be washed or discarded accordingly so that the next user does not come in contact with it. So this is also correct. This is because let's say if some person is handling some biological samples and it got contaminated, then we should either discard it accordingly so that the next user will not come in contact with it. We don't want like any hazardous material to come in contact with the other people who are using the PPE as well. So the correct answer is option D, all of the above. Is this clear? If this is clear, we can move on to the next question. Problem 8. Data acquisition using virtual bench is a more efficient experimental setup as option A, the virtual bench can be used as oscilloscope, function generator, and multimeter simultaneously. Option B, the virtual bench has a programmable power source. Option C, both A and B. Option D, only A. So we also looked at something called as virtual bench, right? So the correct answer is option C, both A and B. The virtual bench is something that like it's an instrument which combines a mixed signal oscilloscope, an arbitrary waveform generator, digital multimeter, programmable DC power supply, and digital input output. It all like is present in one single module, and this module can be connected to a okay. So that is what is called as a virtual bench instrument. So from the given options, we can see that the option A and B are correct. So the correct answer is option C, both A and B. Now let us move on to the next problem. Problem 9. Ideal way to carry fabricated sensors outside a cleanroom facility is option A. Wear gloves and take the sensor by hand. Option B. Use a desiccator. Option C. Use a plastic box or cover to carry the sensors. Option D. seal the sensor using Teflon tape. So which is the correct answer for this question? In this question, we can see like we have completed the fabrication process, right? But we want to transfer the sensor outside a clean room facility. So what kind of method will we use? Okay. So the correct answer for this question is option B, use a desiccator. So what is a desiccator? A desiccator is something that looks like either like this or this. So it has a lid, it has a grease rim. Here is the grease rim. Then it will have a chamber, an upper desiccator plate and a lower desiccator plate. So in the lower desiccator plate, we can keep some silica gel which will absorb any moisture that is present inside the desiccator. And here you can see that there is some kind of outlet as well as wall. So with this, we can pump the desiccator so that it will remain in vacuum. So like from this, you can say that desiccators are generally some kind of sealed enclosures, and we can use to store any moisture sensitive samples or any other samples, critical samples, in vacuum as well as with low moisture. So even though our sensor has been fabricated, like if some, and like one important thing to note is that these sensors are all in micron scale, right? So and like most of the dust particles and those are all in, like they may be several like hundreds of microns large. So if they react with your sensor, then your performance of the sensor will get affected drastically. So to avoid these all, we'll have to keep our sensor in vacuum condition, and like moisture also can affect your sample. So for that reason, we'll keep our samples in desiccator when we are taking it outside the clean room environment. By this way, we can minimize like any interaction of the fabricated sensor with any particles that are present outside. Is this clear? Now let us move on to the next question. Imaging and studying the health and status of cultured cells can be better understood using option A, inverted microscope, option B, compound microscope, option C, stereo microscope, option D, either compound microscope or stereo microscope. So here we see different types of microscopes, and these microscopes can be used for different functions. What we want to image here is cultured cells. So which of this microscope can we use to image a cultured cell? Okay, the correct answer is option A, inverted microscope. Okay, so now let us see how these microscopes will look like. So in this first figure, you can see an inverted microscope. So in this is an inverted biological microscope. Okay. So here you can see that an inverted microscope has the light source and the condenser on top of the sample, and this sample will be pointing downwards. Okay. The objective lens is below the sample. So here you can see the backside of the sample, and in the case of normal sample, we'll have the objective lens above the sample stage. So that is one main difference between compound microscope as well as the inverted microscope. Also in compound microscope, you can see that the condenser lenses in the below the stage as well as the mirror is also there, that is below the stage. So in compound microscope, you can not only sorry, in compound microscope you can not only image samples from the like light from the top, you can also image the samples from the backside as well. So in this figure, you can see a stereo microscope. So in stereo microscope, the main thing is it is also a dissecting microscope. If we look at a compound microscope as well as stereo microscope, the stereo microscopes can generally provide magnification in the range of 10 times to 10x to 40x, and the compound microscope will provide magnification from 40x to 1000x. So this is for generally low magnification, and we have the objective lens above the sample stage. So we'll have light incident uh, from the top, and this will just get reflected off at the surface of the sample, and this is again seen through the eyepiece. So by this you can have a 3D visualization of the sample. So since it has lower magnification, we can also view like large specimens. Like in compound microscope, like the size of the specimen that you can use to see is like smaller than that of the stereo microscope. Okay. So for cultured cells, which is very small, we can use inverted microscope because we need higher range of magnification. Now let us move on to problem 11. Okay. A biosensor has interdigitated electrodes with interconnects spaced at 10 micrometer and contact pads at 20 micrometer. The sensor or readout measurements from these sensors can be obtained using option A, handheld multimeter, option B, micromanipulator, option C, soldering to PCB, option D, none of the above. So which is the correct answer for this question? A biosensor, like how do we measure any particular readout from the sensors? Okay. So the correct answer is option C, soldering to PCB. Okay. First of all, whatever readout of measurements that we are getting, that is electrical. So that is okay. Now, like we cannot use a handheld multimeter. One reason is these multimeters will have probes, and like if you look at the probe tips, the size of the probe tips is very large compared to the size of your contact pad that you are having. So, like we cannot precisely place your multimeter probe on the probe pad. It is very difficult to measure in that. Second option is micro manipulator. So, micro manipulator is like it is an assembly that is just used to like do very precise movements that cannot be achieved with like just human hand. So it will have some mechanism for moving in different directions like X, Y, Z, and also it will have some mechanism for like rotating the sample. However, like unless this micro manipulator is connected to something like a probe, if it is connected to a probe, we can use to uh, probe the sample. Then again, this probe should be connected to some electrical equipment from which we can get the readout. Then only we can use a micro manipulator. Okay, since here it is just mentioned as micro manipulator, we cannot use it. And third option is soldering to PCB. So we can like solder it to PCB and we can directly measure. So if we have soldered it to PCB, which is connected to some sort of readout circuit, then we can directly get the readout or measurements. So the correct answer is option C, soldering to PCB. Now let us move on to next problem. What instrument is used to cut extremely thin, that is 50 nanometer slices of a material? Option A, micromanipulator. Option B, autoclave. Option C, microtome. Option D, molecular scissors. So, which instrument are we using to cut very thin samples of a material? What is the correct answer for this question? Okay. So the correct answer here is option C, microtome. Now let us look at what each of these options are. Okay, first is micromanipulator. So in the previous problem also we saw a micromanipulator. This is how a micromanipulator looks like. So here we'll have these knobs that you see that are for like X, Y and Z motion. So by turning these knobs, you can have like precise movement of the like of whatever that is connected to the device. You will also in some micro manipulators, you will also have a knob for rotation. Okay. So we will have some output section, which is probably this one, where we are having an option to hold a micro tool, and like there are various tools that we can attach to this. Like some of them are like you can attach a micro pipette to it, or an electrode, or a probe. This can be mounted on this, so you can control the motion of the whatever that is attached to this micro manipulator to have like very precise movement, like in the order of microns. You can move as much as a micron at a time, and also you can rotate it as like very precisely. So that is what a micro manipulator is. So we want to cut extremely thin samples. um, we cannot use a micro manipulator generally. Then the second one is autoclave. So autoclave is generally a steam sterilizer, and it is like uses steam with pressure in order to kill any harmful bacteria or viruses that is present inside. So here you can see the structure of an autoclave. It has a lid. It also has a pressure control valve, air removal valve, a thermometer to measure the temperature, safety valve, pressure gauge, and so on. So it will also have some water along with heating elements and a perforated bottom plate. So generally, it uses steam in order to sterilize the material that is present. So here we see the third option, which is rotary microtome. So, or generally microtome. It is a very specialized precision cutting instrument, and with this you can accurately like cut slices from any block of embedded tissue. So let's say if you place your tissue in some paraffin, then you can cut very thin slices, and like there are different types of microtomes are used in order to section paraffin and like plastic which have certain embedded tissues in it. We can also have some microtomes to section any frozen tissues. So here you see that block holder with a block where you can place your block of a sample, and at the base you have a knife holder. So with this assembly you can cut very thin sections of your sample. Fourth option is molecular scissor. So there is something called as restriction enzymes. They are called as molecular scissors because they can be used to cleave DNA at certain specific sites where we recognize. So these sites are called as restriction sites. And what like it does is it will generally cut, or you can say it makes an incision on each side of the two strands of DNA. Uh, so these are also called as restriction endonucleases. So this restriction enzyme is also known as restriction endonucleases. Since here we want to cut a sample, the correct answer will be rotary microtome. Is this clear? If this is clear, we can move on to the next problem. What is the most important article to wear in the clean room? So here we have multiple options. Shoe covers, mask, gloves, buffet, glasses, lab coat, all of the above. So which of all things do we need to wear in the clean room? So this I think we already saw in a previous question as well. We already saw this, right? So let us go through one of the each of the options one by one. So we need to wear shoe covers. We need to wear a mask, gloves, buffet. We also have to wear some protective glasses and a lab coat. So generally it is the correct answer is all of the above. So this is what a buffet looks like. So it is generally a hair cover. So here you have some certain sequence of steps that tell you what are the processes that are to be involved before going into a clean room. So first is you have to ensure that your hands are very clean. Then you will wear some kind of shoe covers, right? You will ensure that whatever the shoes that you are wearing inside the clean room will not have any particles stuck to it. Then you will wear shoe covers on top of your clean room shoes. Then you will wear one pair of gloves. This is then followed by wearing the buffet, the head cover, then your gloves, then your bunny suit, then like your protective uh, shoes over the bunny suit. Then this is then followed by protective goggles and additional layer of gloves. So before you enter your clean room, you again stand on a sticky mat in order to remove any like particles that may be present below your uh, shoes, the clean room shoes. Is this clear? Okay, so what are the advantages of tissue culture? Before that, what is tissue culture? And where do we use the tissue culture? Okay. So tissue culture is a technique where we are growing cells or tissues in artificial environment separate from their parent organization. That is, we are growing it generally in some cultured media. So another name for this tissue culture is micropropagation. It does not just like relate to plants. It can also it can also be correlated with animal tissues as well. And why do we need to do tissue culture? So, in the case of plant propagation, like we can ensure that it will have like desired property that we want. Why do we need tissue culture? So we can do various things like we can produce disease-free plants. We can also do any genetic modification. We can test how the drug metabolism happens and so on. Okay. So if we look at what are the advantages of tissue culture, first is that since we are growing it in a separate medium, it gives us a good control for the enrichment. Okay, so we can like keep suitable culture media where we give the enough nutrients for our particular tissue to grow. The second advantage is that you can do in vitro modeling of in vivo conditions. What does this mean? You can try to create a like in vitro condition that is closely related to in vivo conditions. Okay, so that is one of the other reasons that we can. Then other advantage is that you can characterize, homogenize the sample. Then you can grow like the plants or in like the you can grow the tissue in large scale, which will like lead to reduced cost for growth. Okay. So you can have large scale production of the tissues as well as the cost will also tissue culture. A CO2 incubator mimics the of 37 degrees centigrade, 95% relative humidity, and 5% CO2. Is this true or false? We already saw this in a. Okay, so this is correct, right? It is true. Like in an incubator, conditions that is similar to that of a human body. So we go for neutral pH, then like temperature of 37 degrees centigrade, which is same as human body temperature, and also greater than 90% relative humidity, so 95% is fine. And then we go for 5% CO2. So generally, if you look at the CO2 levels of human, it is slightly greater than 0.04 percentage. However, like why we need to keep this 5% CO2? What H2O will react with CO2? It will react with CO2 to form carbonic acid, H2CO3. It can also react with CO2 to form bicarbonate, HCO3 minus. So this is acidic. Basic. So acidic means your pH increases, sorry, pH decreases. Base means your pH increases. So by controlling your concentration of CO2, you can ensure that the H2CO3 and HCO3 are produced in somewhat equal amount, which will make the pH neutral. And we need a neutral pH because uh, keeping it in a neutral condition will and also our body is mainly maintained in neutral condition. Is this clear? If this is clear, we can move on to the next problem. Problem 16. Autoclave is an equipment used to option A, incubate and grow the tissues. Option B, refrigerate liquid media and growth factors at 4 degrees centigrade. Option C, to sterilize utensils that are used for tissue culturing. Option D, all the above. The correct answer is option C, to sterilize utensils that are used for tissue culturing. Okay, so we already saw the image of autoclave in one of our previous questions, right? In autoclave, microbial life that is present, uh, like there'll be different cycles of it, like water, which is then heated and it is converted into steam. So energy, when water is getting converted into steam, there will be a lot of heat energy that is generated. And like if we are reaching a temperature of around 121 degrees centigrade, then the heat energy that is present in the steam is sufficient enough to destroy a lot of pathogens, including some bacteria, virus, and spores. So here you can see that there is some perforated bottom plate on which we will have some grid basket and pressure instruments or something things to be sterilized. And another important thing is we also remove the so what will happen is whatever heat that whatever steam that is generated when we are heating that why we are doing this is because like if there is some like to prevent the steam from reaching all the parts of the materials which is being sterilized. So that's why we will also have some like air removal steam will reach each and every corner of the uh, equipment that is being sterilized. So we'll have a better sterilization process. And another thing is we also have a pressure control valve. So if we are increasing the pressure, then what happens is the steam molecules will be very close to each other. That means like it will require more energy to remain in gaseous form. So if we are working at normal atmospheric pressure, then what will happen is the boiling point of water will be 100 degrees centigrade. However, in autoclave, we will work at a pressure which is 15 to 20 psi greater than the atmospheric pressure. So pressure is proportional to temperature, so your temperature will also rise. Then your boiling point of water becomes around 121 degrees centigrade in this case. Okay, since your boiling point is also increased, then like at this particular temperature, like it has been seen that most of the microorganisms get killed. So that's why we go for a very high temperature around 121 degrees centigrade. So the correct answer is option C, to sterilize utensils that are used for tissue culturing. Now let us move to next problem. In autoclave, bacteria is killed by option A, boiling in water, B, chemical sterilization, C, high temperature and high pressure steam sterilization, D, radiation sterilization. So this is kind of repetition of the previous question. What is the correct answer? Okay, the correct answer for this question is option C, temperature and high pressure steam sterilization. So like we saw earlier, we like heat water so that it gets evaporated into steam, and the temperature of the steam should be around 121 degrees centigrade. So we have this autoclave working at a higher pressure. So which means that like at higher pressure, the evaporation temperature also increases. So we maintain the pressure at some 15 to 20 psi, which is our atmospheric pressure. So our boiling point of water will also become around 121 degrees centigrade. At this particular temperature, it has been seen that the bacteria and other microorganisms will get killed. In order to ensure that your steam reaches all the corners or nooks of your particular equipment that you are sterilizing, we will also have some air evacuation valve. So it will this will ensure that your steam will reach all the corners of the equipment that you are sterilizing. Now let us move on to next problem. What is the optimum pH required for mammalian cells to survive? Option A 4 to 5, option B 5.2 to 6.2, option C 7.2 to 7.4, option D 12 to 14. So we also saw something related to this in previous questions. So which is the optimum pH that is required for mammalian cells to survive? We know it is neutral, right? What is the neutral range? What is this question? Okay, the correct answer is option C, 7.2 to 7.4. So this is around 7, which we call as neutral. Okay, this is less than 7, so it is acidic. And this is greater than 7, which is basic. So the optimum pH required is 7.2 to 7.4. Now let us move on to next problem. Problem 19. Air curtain is used in a lab to option A, filter the air, B, regulate the particle contamination, C, ensure dust particles do not enter inside the lab while opening the door, D, option E, only B and C. So why are we using air curtain? Okay, so which is the correct answer for this question? An air curtain. Okay. So like for filtering the air, we need some kind of filter like HEPA filter or HEGA filter. So HEGA filter we will use when we want to remove odor as well. HEPA filter is just to remove the particle contamination. So it is kind of mechanical filter. Uh, we use the air curtain to regulate the particle contamination. So that is correct. And we can also ensure that the dust particle do not enter inside the lab while opening the door. So B and C are correct. The correct answer is option E. Only B and C. Okay. So this is how air curtain will look like. So here we are having an equipment where we are having air in a particular this kind of flow manner. So this will ensure that like there are no particles that are entering inside. So it will regulate the like how much contamination that is coming from outside. And also another thing is it will not ensure it will ensure that the dust particle do not enter inside the lab. It does not do any filtering process. It just ensures that there is a flow of air like this, so that the airflow will act like a curtain. So from the name you can see that it is a air curtain. Now let us move on to next problem, problem 20. Pressure inside a clean room is higher than the outside. Is it true or false? So we know that we have different kind of filters inside the clean room to regulate the particle contamination. And also another important thing is that temperature, pressure, humidity inside a clean room are strictly controlled. Is the pressure inside a clean room high or is it lower than the outside? So this answer is for this question is it is true. So the pressure inside a clean room is higher than the outside. So let us see why it is. Okay. So we generally know that air will flow from region of, if you are having a region of high pressure, and if you are having a region of low pressure, then air will flow from high pressure to low pressure. So like another example, one example is that if you are seeing that if you are having a balloon which is filled with air, okay, that means that as you like fill more air inside the balloon, the pressure inside the balloon will start to increase. Once you release the balloon, what will happen is the air from the balloon will try to travel outside, means outside the balloon where there is low pressure. So we like from that we already know that the air will move from high pressure to low pressure, right? So also another important thing is when air is moving from high pressure to low pressure, it does not allow any movement in the opposite direction. Okay, so we are using this concept in clean room in order to stop any airflow inside the clean room. Why do we need to stop the airflow inside the clean room? It is to limit the particle contamination or particle transfer inside the clean room, and also to maintain a cleaner environment. Like since air will generally flow from high pressure to low pressure, it is also easier to remove the like any contaminated air that is present inside for filtering purposes. And how do we achieve this kind of clean room pressure? Like we control how much air is put inside the clean room and how much is withdrawn. Okay, so there are two types of uh, clean room. One is like positive pressure clean room. So here we, let's say this is our clean room. This is having pressure which is greater than atmospheric pressure. So that is our positive pressure clean room. So we will have filtered air that is pumped into the room by using the HEPA filter. And there is something called as HVAC, clean room HVAC system. So what will happen is if we are opening a door in this clean room, since this is a high pressure environment, the air will rush outside. Okay. In case there is some kind of leak or breach in the clean room, then what will happen is whatever air that is present inside, like if there is some door opened unexpectedly, it will move outside the clean room. Uh, but the reverse will not happen. So like it will the contamination will not occur. Okay. So this is how we are maintaining higher atmospheric pressure inside the clean room compared to outside. We will pump in more air compared to the like by means of this HEPA filters, we will pump it consistently into the room at a rate which is lower than the evacuation. Okay. So if you want to maintain a positive clean room, or sorry, if you want to maintain a positive pressure or negative pressure in clean room, we have to have some adjacent space which is maintained at a different pressure. So let's say if you want to maintain this at a pressure greater than atmosphere, then we will have a adjacent space which is maintained at a pressure which is at atmosphere pressure, uh, so that we can just balance the pressure of the entire system. So now let us move on to the next problem, problem 21. An impedance analyzer can be used to measure option A, mechanical impedance, B, electrical impedance, C, both A and B. So which is the correct answer? This is an impedance analyzer, and we saw that we had one virtual bench, right, which has impedance analyzer and so on. So what is the correct answer for this question? The correct answer is option B, electrical impedance. Now let us move on to next problem. A virtual bench can be used as option A, multimeter, Option B, oscilloscope. Option C, power supply. Option D, function generation. Option E, all of the above. So this already we saw in previous question. What is the correct answer for this question? So the correct answer is option E, all of the above. Okay. Because we already saw that it can work as a multimeter, oscilloscope, a DC power supply, and also as a function generator. So any questions? If not, we can end the session. Thank you.